Okay, hello class. So this is the last part of the series of protein turnover and amino acid catabolism. You'll see a bunch of reactions in this presentation. Don't worry about actually like memorizing all of these things. I'll highlight what's important to take away, and it's more of a reference point and general education. So, for today's topic, we will talk about carbon atoms of degraded amino acids emerge as major metabolic intermediates, and then very briefly, inborn errors of metabolism can disrupt amino acid degradation. So we have a lot of different major intermediates that happens within our metabolic system. And so they try to break down where some of the degraded amino acid, the carbon atom structure, where it can generate these different intermediates, such as the acetoacetyl coenzyme, acetyl coenzyme, the alpha ketoglutarate, the succinyl coenzyme. They're just highlighting the enzymes that are degraded in order to form these key intermediates. And so you'll start to see some are highlighted in yellow, and then some are in this reddish pink color. And so what it's really trying to say is these amino acids are called in term ketogenic amino acids because it can give rise to ketone bodies or fatty acids. And then as far as the ones in red, that is in term called glucogenic amino acids, and that is because they can be used to produce glucose. Now, some are actually both ketogenic and glucogenic type of amino acids, and they are listed here: isoleucine, phenylalanine, tryptophan, and tyrosine. But there are some that are only ketogenic, and that's the leucine and lysine. And the other 14 amino acids are classified as solely glucogenic amino acids. So talking about some of the breakdown of some of the intermediates, we'll go through several reactions. And so as far as tryptophan can be broken down into aniline, which broken down to pyruvate, and you see that it is highlighted where is that carbon structure that that correlates to the formation of this pyruvate. And then you have glycine that can be degraded down to serine, that can be degraded down to pyruvate, and cysteine can also degrade down to pyruvate. And so you have this aniline that goes through a transamination with alpha ketoglutarate to form that pyruvate that I mentioned before. And it also produces glutamate. This should be very familiar if you looked at part 2 of how amino acids being transferred over to an alpha keto acid to ends up forming these glutamate. And in this case, it could produce pyruvate. And then you have the serine gets produced to pyruvate, and you have the ammonium ion that is produced. So it's pretty much a release of the amino group, and then also you have a release of that hydroxyl group as well. But this can happen in several pathways, so there's not a single path. But just note that pyruvate amino acid the carbon structure from that serine amino acid can be used to make pyruvate. And then also just remember that glycine can be converted to that serine, and then from there to pyruvate. Another intermediate called the oxaloacetate that can be produced from aspartate amino acid, where the amino group is transferred over to alpha glutarate. And if you ever see that alpha ketoglutarate, we know we're making glutamate. And the other product is that oxaloacetate. And so, as far as the glutamate, that can be formed, that carbon structure can be broken down from glutamine, proline, arginine, and histine. And remember that glutamate can always be go to that dehydrogenase to release that ammonium ion to form that alpha ketoglutarate. And so this is just a basic knowledge of that histidine going through different reactions to end up forming the glutamate. You do not need to memorize this reaction. Just use it as a source of reference. Same thing with this one, but just kind of know that proline and arginine, they both gets broken down to glutamate, gamma semialdehyde, and then from there it gets produced to glutamate. Then we have the succinyl coenzyme, gets broken down from these listed amino acids. And so one of the most important ones is methionine. It actually produces what's called SAM for short, which is S-adenosyl methionine, and that is actually a methyl donor in our cells. And so it goes through, once again, all of these several steps that you do not have to memorize this, be able to follow it. And then you have the succinyl coenzyme A. And then you have leucine, that gets broken down as well, go through this transferase, where that amino group gets removed, and then the coenzyme A gets attached to the carboxylic acid group to form this structure. And then from there, we're going to eventually get to the acetoacetate and propiononyl coenzyme, but it goes through some other steps that you don't need to memorize, but it ends up getting to that acetoacetate and acetyl coenzyme. So this more so generally know that branch chain amino acids yield acetyl coenzyme A, acetoacetate, and propiononyl coenzyme A. And then other thing to just kind of know in general, that aromatic amino acids are broken down by molecular oxygen, and then from there it eventually produce fumarate, and that is this. For this case, it was fumarate because it's phenylalanine, but just know in general the aromatic amino acids are broken down by molecular oxygen. Same thing with here, where you have tryptophan, was broken down with the molecular oxygen and go through several steps to end up making acetoacetate. And ending things with inborn errors of amino acid metabolism, when you have certain diseases it causes certain enzyme deficiency and produces these symptoms. Once again, this is more so for general education and used as a reference. And that is all for the third part of the series.